गुड मॉर्निंग फ्रेंड्स। आप सभी को मकर संक्रांति की हार्दिक शुभेच्छा। और मैं आशा करता हूँ कि एवरीबॉडी इज डूइंग वेल विद गुड हेल्थ एंड स्टेबिलिटी। टुडे बिफोर गोइंग टू द मेन टॉपिक that I have come in front of you, that is my second part of the topic blood. I want to discuss in front of you regarding stem cells, little bit about stem cells. थोड़ा stem cells के बारे में हम लोग जानकारी ले लें, because in the present topic or the topic that I am going to represent or present in front of you, the concept of there should be some little knowledge regarding stem cells, what is stem cells and what is functions inside the human body or in the human body. So stem cells are such kind of cells that are present in the human body and they are capable to create new cells. Okay, right from the muscle cells to the brain cells. They are also helpful to repair damaged organs. Research is going on around the world on the concept of stem cells, and the scientists around the world are trying to find a solution on various kinds of dreaded diseases that is prevalent in the society, such as diabetes, Alzheimer's, cancer, neurodegenerative conditions. In all these conditions, the scientists are trying hard to get a proper solution so that a person get relief from the disease process and lead a very normal life. I hope that in the long run we will get a good result with the help of stem cells therapy, and it will be very helpful also for the families that they are guiding or that they are protecting or doing hardships with the patient, because when a disease patient is present in a family, the person who is having such disease, not only that person suffers but also the family also suffers. So stem cells therapy, I hope, will bring a new ray of hope in the future process, so that the disease process will decline in the long run. So now let's go to the my topic regarding blood, and let's see that how a blood cells begin their life. How a blood cell begin their life in the human body. The blood cells begin their life in the human body with the help of a single cell, that is pluripotential hematopoietic stem cells. Pluripotential hematopoietic stem cells are such kind of cells that are responsible for the development of or production of different kinds of blood cells that are present in the human body, and such kind of blood cells are neutrophils, eosinophils, monocytes, lymphocytes, RBC, platelets. All these kinds of cells are developed from the pluripotential hematopoietic stem cells. Then these kinds of stem cells are responsible for the growth and development of the all kinds of blood cells in the human body. When such kinds of cells are present or reproduced in the human body, then some part of it they remain in the bone marrow. In my last topic we have discussed regarding bone marrow little bit. They reside inside the bone marrow, and whenever the body is in need of such kind of cells, they used to generate or they used to give proper stimulation to the bone marrow, and with a certain process only such kinds of cells they used to grow inside the in the blood, or whenever the body is in need, they used to take it from the bone marrow through a process. There is a process for the erythrocyte development, and I will discuss in the later part of this video. And so simply you understand this, that whenever the body is in need of a cell, they used to take it from the bone marrow, because the body physiologically it used to keep such kind of cells as a reservoir in the in the bone marrow. Now before approaching further, I want to discuss in front of you regarding what is progenitor cells and what is committed stem cells. So progenitor cells, actually these kinds of cells are the descendants of the stem cells. Descendants means they are developed from the stem cells only. So these are the descendants of the stem cells and they are present all over the body, and these kinds of progenitor cells are responsible for the development of a cell of a particular tissue or organ. Is it clear or not? That progenitor cells are responsible for the growth and development of a cell of a particular tissue and organ itself. They will not produce all kinds of cells, but tissue specific or organ specific. Committed stem cells are more or less same only. Committed stem cells, these kinds of cells are committed to a particular tissue or organ or for particular line of cells. So for that reason, they are known as committed stem cells. So progenitor cells and committed stem cells are more or less same. Okay. Then when a committed stem cells are grown in a cultural media, they give rise to blood cells. The committed stem cells that are responsible for the development of erythrocytes, they are abbreviated as or are designated as colony forming unit erythrocyte. Such kind of designation is also given to granulocytes and monocytes, such as colony forming unit granulocyte, colony forming unit monocytes. These are the examples. Actually, committed stem cells, if they will produce granulocyte, they are known as colony forming unit granulocytes, and if they produce monocyte, they are known as colony forming unit monocytes. Simply, and again, if they will produce erythrocytes, they are known as colony forming unit erythrocytes. These are simple designations that are given to the particular committed stem cells or particular cellular formations. Okay. Now, the growth and development of a stem cells depends upon some kinds of protein that are present in the human body, and these proteins are known as growth inducers. These growth inducers are responsible for the growth and development of a stem cells. 4 kinds of growth inducers are hereby described, but one among them is interleukin 3, and this interleukin 3 is responsible for the growth and reproduction of almost all kinds of committed stem cells, and other 3 kinds of proteins, they are responsible for the growth and reproduction of the specific cells only. But interleukin 3, these are responsible for the growth and development of all kinds of committed stem cells. Now, growth inducers are only responsible for the growth of a stem cell, but they do not play any kind of role in the differentiation process. This differentiation process is usually controlled by another group of protein, and they are known as differentiation inducers. They are known as differentiation inducers, and these differentiation inducers, they differentiate committed stem cells from multiple times and ultimately mature blood cells or red blood corpuscles they used to, or mature blood cells they used to form. Formation of growth inducers and differentiation inducers are usually controlled by some kind of factors, and these kinds of factors occur outside the bone marrow. With the help of these factors, the formation of growth inducers and differentiation inducers are usually controlled. Okay. Then let's go to the next topic, that is stages of differentiation of RBC. What are the stages of differentiation of a RBC? Proerythroblast is the first kind of cell in the blood series. Is the first kind of cell in the blood series, that is proerythroblast. Under proper stimulation, when a stem cell produce proerythroblast, they divide repeatedly and ultimately mature RBC is produced. Mature RBC or red blood corpuscles they used to develop from proerythroblast. that is proper stimulation should have to be done. Okay, this is a physiological process, and after that physiologically proper stimulation is happen, and ultimately from the stem cells the pro-erythroblasts develop. Pro-erythroblasts they used to develop, and they divide multiple times, and ultimately mature red blood corpuscles they used to the development of red blood corpuscles occurs in the human body. Again, the first generation cell is known as basophil erythroblast. The first generation cell of the blood cell series is this one, basophil erythroblast, and in this basophil erythroblast the hemoglobin content is little, the hemoglobin concentration is little, and this basophil erythroblast they divide and ultimately polychromatophil erythroblast formation occurs, and again polychromatophil erythroblast divide and ultimately orthochromatic erythroblast formation occurs. Then in the succeeding generation, in the succeeding way, the concentration of RBC in the cell increases to 34%, and the nucleus also become extruded or become smaller in size, and the remnants they used to get absorbed or are thrown outside the cells. And at this stage, the cells are known as reticulocyte, and in this reticulocyte basophil erythroblast remnants are present, mitochondria remnants are also present, Golgi apparatus remnants are also present, and cytoplasmic organelles are also present. Okay, now in this reticulocyte stage, the cells come out from the bone marrow and they reach the capillary through a process called diapedesis. Diapedesis means squeezing through the capillary membrane, and ultimately within 1 or 2 days in the reticulocyte stage the basophil contents they used to disappear, and ultimately leads to the formation of a mature erythroblast. Mature erythrocyte development occurs. So let us summarize in this video that what we have come to know. We came to know that we discussed regarding little bit about stem cells and what kind of research is going on around the world on stem cells, and we also came to know that how blood cells begin their life, how blood cells used to develop from the pluripotential hematopoietic stem cells. Then we also discussed regarding committed stem cells and progenitor cells. Then also we discussed regarding growth inducers, differentiation inducers, and what are their roles inside the human body. And also we discussed regarding the stages of differentiation of RBC, that is how mature erythrocyte development occurs, what are the stages. We discussed regarding pro-erythroblast in this topic. We also discussed regarding basophil erythroblast. Then also we discussed regarding reticulocytes, and ultimately erythrocyte formation occurs in the human body. These stages are very important for the normal functioning of the human survival, or for normal functioning of all the organs, because any kind of deformity in this process will lead to development of different kinds of diseases in the human body, which will be discussed in the later part of my video. I hope the video that I have presented today will be fruitful and will be helpful for my friends, and I hope more and more comments or more and more suggestions that I will receive from my friends and all. And if my friends didn't like till now or didn't subscribe till now my video, then I am requesting everyone to kindly like, comment and subscribe my video. And within few days I will again come in front of you with my with the third part of the topic blood. And for till then, goodbye and Shubha Din. Thank you.